it's my pleasure to introduce Dr. Alpha Lee. When I was a postdoc, he reached out to us with this really cool idea about how to use random matrix theory to come up with a baseline for one of our models that we were doing for ligand-driven drug discovery. And then that next summer, I met Alpha at one of the Gordon Research Conferences, and we hit it off, and it was just such a flurry of ideas, and I really enjoyed getting to know Alpha. And I've been following his work closely. He's was at Harvard and now has been in Cambridge with his own group. And recently he's been running, as he says, a drug discovery biotech company in the open, publicly. So we'll hear about this COVID Moonshot project, which is really innovative approach to collaborative drug discovery. And he's got some really exciting ideas about combining machine learning and structure and ligand-based drug discovery. So I'm thrilled to have him a chance to come and present to us. And for the graduate students that are here, you're invited to stick around for an extra 30 minutes after to have a conversation with Alpha. So please join me in welcoming Alpha to present today.
Yeah, thanks so much for the invitation. Pleasure to meet you all virtually. My talk today will be about the COVID Moonshot initiative, which is an open science initiative trying to find SARS-CoV-2 main protease inhibitors with a crowdsourcing open approach. And I will first talk about Moonshot and our progress. And then the second part of my talk, I will focus on specifically diving into how machine learning has aided Moonshot and some learnings we have got from applying machine learning in a very prospective setting in the open, trying to find an actual drug. Before I talk about Moonshot, let's take a step back and ask the question, well, why do we need an antiviral against COVID, seeing that, vaccines are now available? Why do we still need to do drug discovery? The reason is that we think antivirals are operating in a very complementary way to vaccines. First, vaccines are known to not be completely effective. In fact, there's age groups which may not be which may not be appropriate for vaccinations, and uptake still leaves a big problem. More importantly, orally available pills are often much easier to make, much easier to distribute, much easier to administer. And antivirals have a large window of opportunity to work when a patient develops mild to moderate symptoms. It is known that the end stage of COVID usually occurs with immune overdrive, whereas where there's a clear window of opportunity when the patient develops a mild to moderate illness, when antiviral could be very useful. And more importantly, antiviral can also be used in a prophylactic sense, administered to patients who has been in contact with people with other confirmed COVID positive patients. So we think antiviral, in particular, orally available antiviral, plays a very important role alongside with vaccines and public health intervention in combating this pandemic. So focusing specifically on therapeutic intervention against COVID, if you take a step back and look at all the viral mechanisms, one protein particularly stands out as an eminently druggable target, and that's the main protease. The main protease, in a nutshell, enables the virus to replicate itself, and inhibiting the main protease thereby shuts down viral replication. And therefore, trying to drug and target the active site of the main protease, or Mpro, is a very viable strategy to go after COVID. Moreover, the main protease is also distinct in sequence to any host human proteins. Therefore, going after the main protease minimizes the risk of unintended targets and off-target effects. And so this is going after the main protease is definitely not a new idea. Even back in SARS-CoV-1, there's been a lot of work targeting the main protease, establishing the mechanistic relationship between a main protease inhibitor and viral death, and to some extent even in vivo models of a viral infection, showing that there's a very strong disease target linkage. What is even better about the main protease is that it's actually a target, a protein that is broadly conserved across many pathogenic coronaviruses, SARS-1, MERS, COVID, and many other coronaviruses. Actually, if we align the sequences, we realize that the main protease itself is broadly conserved, which means that having an antiviral against COVID might mean that this antiviral could be broad spectrum. And the structure of the main protease was released back in February of last year, revealing that the binding site actually shows remarkable similarity with SARS-CoV-1. In fact, the drug that is currently under clinical trial for Mpro inhibition produced by Pfizer was actually a program for SARS-CoV-1 back in 2003. They just filed a patent and shelved it until now. However, apart from the drug that I mentioned, currently under clinical trial by Pfizer, there is actually no human coronavirus Mpro inhibitor approved as a drug. However, there are drugs for cats for main protease. The feline coronavirus is a very similar coronavirus to, in terms of Mpro sequence identity, to the human SARS-CoV-2, and there is a compound in late-stage clinical investigation for cats. So again, another piece of evidence that there's a strong target disease linkage, and Mpro is a eminently druggable target. However, looking at the many different drugs, clinical candidates, and advanced clinical candidates currently undergoing investigation, there are two common themes which emerges. The first is that they are peptidomimetics, which means that they look like they are very big molecules and peptide-like. The second is that a lot of the compounds work via a covalent mechanism, trying to form an irreversible covalent bond or reversible covalent bond with a cysteine residue in the catalytic site. In fact, the Pfizer clinical candidate, which I alluded to, is both a peptidomimetic and a covalent compound. And peptidomimetics are notoriously hard to develop into oral therapeutic. And not surprisingly, the Pfizer drug, or the Pfizer clinical candidate, I should say, is an IV drug. The same with other COVID medications like remdesivir. So there's an acute need to develop a orally available small molecule drug. And more important, and in addition to that, the covalent mechanism that a lot of these drugs operate in does raise the concern of idiosyncratic risk down the line. And so another desirable feature is if we can develop a small molecule, non-peptidomimetic, orally available drug that operates on a completely reversible mechanism that appears to be the quickest and safest way that we can get to an oral antiviral. And so that, in a nutshell, is the premise of Moonshot, trying to find a orally available small molecule antiviral. The whole project started back in, I guess, February 14, 2020, almost a year ago, or actually just a year ago. The main protease was cloned and produced by Martin Walsh in Oxford. Then Frank determined the atomic resolution of the structure in February 20th, I think replicating some of the work done by a Chinese group. Then near London and Weizmann did a covalent screen finding hits in February 25th. And then March 5th, they on a single day, they collected 1,500 co-crystals. And in March 18th, they released the crystal structures on PDB and on the Diamond website. So all in all, in just over a month, they went from cloning the protein to doing a fragment screen. And the fragments completely cover the active site, which is beautiful. here is a snapshot of a zoomed in view of the active site and the fragments that decorated the active site, showing that different regions of the active site, the different pockets in the canonical nomenclature, the P1 prime, P2, P4, and P1, including here, the yellow box shows the region where the catalytic cysteine residue, which is a residue primed for a covalent mechanism, are densely populated by fragments. And that seems like a treasure trove if one were to think about developing something out of that, because there seems to be a lot of opportunity for fragment expansion and fragment merging. The fragments also bind very deep into the binding site, meaning that there is actually quite a lot of mileage one can gain from just looking at these fragments. And here's another zoomed in image showing that a lot of the fragments just overlay beautifully onto each other, again teasing us with the idea that maybe fragment mergers or fragment expansion could be a way where we can rapidly get to potent inhibition. As I said, all the data were released by Martin and Frank and Nia and others directly online. And back then I was actually in California, starting my startup Postera. And we looked at the data and we looked at Martin's tweet actually. I was on Twitter and looking at that, and I was like, wow, that's like great. And one of my co-founders were like, let's really help drive this effort from a fragment screen and a structural biology work into potentially a drug against Mpro. And we were really emboldened by this idea and the open science ethos. And so we want to take it one step further and actually launch a drug discovery effort out of these beautiful structural biology data. And so Moonshot was born almost a week later. The premise of Moonshot is, well, there are a lot of fragments out there that Frank and others accumulated and released. Why not release this to the medicinal chemistry community and like-minded colleagues and ask them, out of these fragments, how would you merge them together from a potent inhibitor? And what are the fragment expansion ideas that you would suggest us to do? And we literally have a website with a catcher with a chemical drawer and ask people around the world to chime in, look at the structures, and draw their favorite compound and submit. Seems like a very naive idea, but we thought, well, why not? Back then was the first wave of lockdown, and we hoped and reckoned that a lot of medicinal chemists are probably working from home, and hopefully they would have some time to chime in. And Nature Chem also kindly ran a correspondence piece for us, advertising this effort to the world scientific community. We thought, trying to draw molecules from fragment merger seems like a rather niche activity. Wouldn't say it's necessarily the pastime of most people. So we thought maybe we would get 100 submissions, and as long as those submissions are great, like that should be at least a good starting point. That as an initiative we can think about how to build on that and actually nucleate the community. But we were incredibly humbled and emboldened by the response that we got. To date, we now have 16,000 designs submitted to our website from 450 designers around the world, and that includes a lot of very experienced medicinal chemists from the industry. In fact, UCB Pharma joined the consortium, and they dedicated 10% of their time, their MedChem design time, in helping us come up with ideas. And a lot of retired chemists from the likes of Pfizer, AstraZeneca just freely joined and gave us their ideas and their insights. To date, we have synthesized and tested over 1,700 compounds against Mpro. I would talk a little bit more about the synthesis part and the testing part later in my talk, and learnings that we gleaned from making so many compounds. So now there is a lot of compounds we have established. Like even after the first week, we got like 2,000 compounds submitted after the first week. So we established very quickly that there is actually a lot of interest in helping us, and we were really emboldened and humbled by the support from the community. But then, as you can glean from the statistics, we can't obviously make all the compounds, and we in fact made only around 10% of it. And so the question becomes, we now have 2,000 compounds, like what do we do with it? And how do we decide? And how do we rapidly triage so many compounds to figure out which one shall be made? And note that back then, it was a year ago, we were still very much in the fragment to hit stage. The probability of a compound being a hit is on average pretty low. So we're trying to make as many as possible. And that's where a machine learning technology, which I would take a deep dive later on, becomes incredibly useful. In a nutshell, I will talk about that later in the talk. We use machine learning to design synthesis route at scale. So basically, we take all the crowdsource ideas, put it through a machine learning algorithm. The algorithm connects to our partner contract research organizations like INAMIN, WuXi, and Sai Life Sciences in India, and automatically designs a synthesis route. And then we know exactly how hard it is to make a molecule in a particular CRO, so that we can say, hey, this is a one-step route, two-step molecule, three-step molecule. And then in the first round, just say, whatever that can't be made in 2 steps, well, we are going to wait till another day to make it. And that allows us to rapidly turn the synthesis wheel. I would talk about this a little bit more later in the presentation. But just to say that this effort has spiraled into a global collaboration. We literally, our collaborators actually span most of the world, ranging from virology in Israel to structural biology in Oxford, folding at home, which is the world's, I think at one point became the world's only exascale computing resources, because so many users wanted to chime in and contribute and donate the CPU time to folding at home. And so we, for a few months, I think, became the world's largest supercomputer, running free energy calculations, etc. This map summarizes only part of our, only some of our collaborators, and we are really, really fortunate to have a lot of people willing to work for us pro bono. And this allows us to establish I guess, a relatively robust assay cascade, going from enzymatic biochemical assays done in actually 2 places, Oxford and Weizmann. So we do 2 assays for every molecule in 2 different methods, fluorescence and rapid fire, cytotox and crystallography. That's done weekly. And then compounds are then, promising compounds are then ran in live virus assays, again, ran weekly in Israel. I think that input is really useful, having weekly turnaround in viral assays. And then tier 1 ADME, PK, and then right now we are at the PK stage. And beyond that, we have also aligned collaborators to ascending dose tolerability, efficacy studies, and other diagnostic assays. And so this cascade, at least the ones highlighted in green and yellow, is running on a weekly basis, where we cycle compounds through the different assays depending on whether they pass potency threshold. And I think this open science approach, where all the data is being released immediately and we claim no IP and no patent, means that a lot of people, very kind contributors, are in a position to collaborate with us to allow us to establish this assay cascade, which generated a lot of data, which I will go on to talk about later. So you may ask, well, how does the crowdsourcing actually work, and has the crowdsourcing actually yielded anything? And this is an example of a submission actually from that website I mentioned a few slides ago. We launched a website, Moonshot, and then asked people to submit things. This is one of the first submissions actually that we got. A person, Tryphon Sarganis in Oxford, basically looked at the fragments and proposed the following merger shown in the box. He literally said, let's merge these 4 fragments together. And he thinks this compound triuni 714A76B6 would be an interesting compound, and actually purchasable from MQ and Mpro, so he bought it. And this compound has an IC50 of 23 micromolar. Not bad, seeing that they're starting actually just from fragments, right? It's not even we are not into HTS, it's just fragments. So that sounds like an interesting hit. And this is an example of a hit that we got from the crowd. And the whole Moonshot project is completely open science, where we release all data the minute we receive it back to the website, and to solicit more people submit and ask more people to submit things to and ideas to us. And this cycle has continued now many times. I think we are now on our 40th week or something of assay results. And so this iterative data release, crowdsourcing cycle it's converged onto 4, I think, salient non-covalent series. The aminopyridines, the UGIs, the quinones, and benzotriazoles. 1 primary series and 3 backups. And the 4 series on so 2D view looks very different. They have very different chemotypes and bond connectivity, but gratifyingly, they actually share a very similar mode of binding, as in they span similar regions in the binding site and basically fills the binding site pretty well. And that's nice because we have now many different chemical matter and chemotypes that spans the binding site. And obviously different chemotypes would have different ADME profile and then tox profile. And so having all these diverse starting points and chemical matter allows us to make progress in understanding whether we can graft one chemical matter from one series to another and vice versa. Just give you an example of how this iterative crowdsourcing driven lead optimization has worked. So I mentioned this person in Oxford submitting the first compound. It's a literal, we call it aminopyridine because it was synthesized from an aminopyridine coupled with a phenylacetic acid that was 24 micromolar. We then immediately released it back to the crowd, and sometime later, folks at UCB, a pharmaceutical company, submitted this compound going from methyl pyridine into an isoquinoline that is now significantly more potent. And then it was suggested, why not cyclize it to form a benzopyran? This gives a little bit more conformational rigidity. Again, now the potency has improved. Do a chiral resolution, lo and behold, it improved 2x. And finally, adding a methoxy in the quaternary carbon gives now an 80 nanomolar inhibitor against the main protease. And this 80 nanomolar inhibitor is actually also active against live SARS-CoV-2 virus with a 500 nanomolar EC50. And at every stage of the process, we have rapid structural biology feedback so that we actually guided by and enabled by very advanced structural biology insights and inputs. This project is obviously not about ligand discovery. We are about drug discovery, and we have also addressed ADME problems in lead optimization. For example, the first benzopyran compound that we got has a relatively low solubility and a relatively low half-life in human liver microsomes, and we have been able to rapidly navigate and around this chemical space, showing that if you turn a benzopyran into a tetrahydroquinoline, that improves both solubility and human liver microsome stability. It essentially forms an extra, it has an extra hydrogen bond donor, and therefore not surprising, it reduces the log P. And interestingly, if you just add a chlorine there to the phenyl ring, you also improve metabolic stability. And the punchline of that is that we have now found actually multiple ways to maneuver in chemical space where we essentially retain potency, but we can navigate the ADME space in a very facile way. In terms of where we want to be, we actually have a target product profile of where we want to hit. And the column on the left are sort of our key target product profile in terms of high potency against the biochemical assay, good antiviral properties, important PK/PD properties. We have a Cmin above IC50 for at least a day, and then hopefully a broad spectrum, active against a broad spectrum coronavirus. Definitely want to be oral, want to be a class 1 from biopharmaceutical, so we aim for high solubility, a long half-life, and sort of the standard safety pharmacology requirements. And progress to date, we are almost meeting the protease activity and viral replication goal. we are still in the process of doing a set of mouse PK studies and corona spectrum studies to understand the PK properties. We have actually got compounds with oral exposure already, although the half-life can definitely be improved. Solubility is doing well. In terms of safety, because the compounds are extremely small, they're actually very, very ligand efficient. And so compounds are pretty clean in protease panels and also pretty clean against safety pharmacology panels. And I would emphasize that perhaps it is a consequence of the ability of nucleate a lot of ideas from disparate sources from the crowd. We have actually converged onto a set of compounds that is extremely, extremely ligand efficient, in the sense that a very, very small compound leads to I would say, surprisingly high potency. If you compare it against preclinical candidates based on peptidomimetic, those are pretty big molecular species, whereas here we are dealing with a really small compound yielding a nanomolar, a low nanomolar antiviral inhibition in the enzyme assay and a nanomolar inhibition antiviral assays. So I think that gives us a lot of hope and promise that actually these we are landing onto chemical matter that can be rapidly developed into a promising drug. And here's a leaderboard of our current top compounds. The first row is the benzopyran, which I alluded to is not soluble and rapidly cleared. We have now solved the clearance problem in several ways. And as you see now, we are in a regime where we are landing compounds that have reasonable half-life, reasonable solubility, clean off-target effects, and clean and no selectivity problems with decent, hopefully decent in vivo PK, although that's something that we are now rapidly gathering data for and exploring now the PK SAR space. And again, the compounds are still very, very small and ligand efficient. So that's the sort of where we are. And the next part of the talk, I want to take a step back and I guess reflect a little bit on now we have ran the design make test cycle for 40-something cycle. What is the rate determining step? I think there's perhaps a lot of excitement in the literature on potency prediction and predicting bioactivity of ligands and designing better ligands. But if we sort of take a step back and just chart the number of calendar days in each part of the design make test cycle, what surprises me is that the design stage, we typically would have an hour-long meeting every week talking about compound design. People go back home, they can do machine learning or docking or FEP. Max would be a week to design a compound. I mean, if you just do standard SAR, then you can just draw a compound and that would be like hours, right? Synthesis, on the other hand, even a very simple compound would take like 2 to 3 weeks to make and just purify an LC-MS and all the sorts of fun stuff they need to do to confirm the identity of the compound. So that can be 3 weeks, or you can even go up to 8 weeks if you're unlucky, right? And even then, you may have to drop the compound because you really can't make it. Testing is high throughput, biochemical assay. Viral assay is a bit of a pain. You need to go to BSL-3 lab, so a day to a week. Even PK like studies, right, can be done in two weeks. Shipping is the amazing bit. I've never respected DHL more in my life. Before being involved in Moonshot, it's actually a time sink. If you run it over and over again, it's amazing how fast we have gotten shipping to. I guess from the good old days of weeks to now less than a week, but still it's like 1 week of the design make test cycle. So the take-home message is that actually synthesis is the rate determining step if you think about design make test cycle. Now, obviously one could argue that if we can design the best compound, then you can just make the best compound and that would be the end of it, rather than going round and round and round. However, if we think about it, if you make a wrong compound, or we don't know whether a compound is a 2-3 week compound or an 8 week compound, that is like a 2 to 3x loss in time. If you want to compensate it in the design stage, then you really need to improve your enrichment factor by a lot to compensate that. And so an incremental or even a statistically significant improvement in, let's say, enrichment factor AUC or whatever metric we use for potency prediction, that's great. But if you can't accurately forecast whether a compound can be made quickly, then you just sink a lot of time for relatively little benefit. And so that's one of the, I think, take home that we got from running this many times, is that actually synthesis is incredibly important. And it's actually one of the, if not the rate determining step, and trying to reduce synthesis by improving quality of design, in theory would work, but you really need the design to improve by a lot compared to the baseline, which is, as standard SAR or docking or whatnot. So trying to achieve that delta is hard. But what we find actually, which I will go and talk about in the second part of my talk, is that trying to shave off time in the mix stage is relatively achievable with machine learning. In particular, trying to have a very accurate estimate of whether your compound is easy or hard is something machine learning can definitely help. So I'll focus on the mix stage for the remainder of my talk, because this is one of the fairly painful stages of this process, and it's a stage where we think machine learning can actually help in accelerating the whole cycle. So again, why is synthesis important? Well, synthesis is important not only in the abstract sense, it takes a long time, but also because it's a large variance in time it takes. For example, the compound on the left is one of our first benzopyran hit. It is a bicyclic. It has a bicyclic structure, the benzopyran. It looks relatively complicated, but it's actually a one-step synthesis. It takes enamine a week to make this and purify this, another week to ship. The compound on the left is a slightly different configuration of a 6-membered ring with an oxygen there. This beast took 2 months to make, and we almost failed to make it. Only with a lot of attempts, we can actually make that compound. And so relatively complex, the take home of this observation is that relatively complex compounds, complex looking compounds can be very easy to make if you have the correct inventory, and relatively, and compounds which have the same complexity could actually then be astronomically hard to make, because you just have several steps that turns out to be a minefield. And the slide that I put there with rats, that's like attempts failed, attempts failed, attempts failed. So being able to forecast these minefields, that's very important. So how do we do that? Well, let's take a step back and think about like, how should we accelerate synthesis using machine learning? Well, one way to accelerate synthesis using machine learning is, well, if we can predict at least the outcomes of chemical reactions accurately, well, that would avoid a lot of these rat failures shown in the slide, and that might help, right? And then we go one step further and start planning synthetic routes. So then, okay, let's answer the first question. Well, how do we use machine learning to improve our prediction of chemical reaction? And that leads nicely into trying to learn what I would call a language of chemistry, this machine translation approach to reaction prediction, which we developed in the last couple of years. The idea is basically that rather than thinking about necessarily sketching out the mechanistic steps in a chemical transformation and trying to, infer from the arrow pushing in Orgel classes or DFT calculations if you're doing theoretical chemistry, why not lean on the vast amount of reaction data that is currently available, which you can just basically download from patents, and just, treat a chemical reaction as machine translation, where the reactants and reagents are the input and the product is the output. And you can just, effectively Google translate your way to reaction prediction. That seems like a very naive idea, but we actually showed that this really a naive model of the effectively natural language processing approach to reaction prediction has a tested accuracy of over 90% on a data set of reactions available in US patents, and is 10% more accurate than the best human experts when it's benchmarked against a data set released by an MIT group. And more importantly, the model not only knows the correct product, so given reactant and reagent predicts the product, but the model also returns an uncertainty score. And it turns out that using this uncertainty score, we can predict whether a prediction is right or wrong. So the AUC of that prediction is 89%. In other words, the model is 90% accurate in predicting the correct product given reactants and reagents, and 89% correct in predicting when its own prediction will be wrong. That's all numbers. To give you some chemical context of what that means, concretely, the model can predict challenging transformations like chemoselectivity. We can see here are two amides, which one get reduced first. The original paper find it challenging to predict. The model correctly predicts that. Stereoselectivity is a standard diastereoselective reduction. Which phase is the hydride being delivered on? The model correctly predicts that as well. And here, there's a competition between Baeyer-Villiger reaction and epoxidation. The model again correctly predicts that. So that's sort of some examples from the literature. You may ask, well, these are selected examples perhaps. Is there like a general way to quantify or qualify the power, the accuracy of machine learning-based reaction prediction relative to some baseline? And so we took a data set actually published in 2018, where the authors tried to predict the outcomes of electrophilic aromatic substitutions. That's a notorious class of selective chemical transformations where the goal is, for example, here is a bromination. The goal is to predict which carbon-hydrogen bond will get brominated. There are, and in top molecule, for example, there are 5 aromatic carbon-nitrogen bonds. So there are 5 possibilities, and experiments may observe one product. And the question is, can we predict which product should be formed favorably? And the authors in that paper used semi-empirical quantum mechanics, and we showed that molecular transformer effectively achieves the same accuracy than semi-empirical quantum mechanics, and obviously incurring a fraction of the computational cost. So how does that actually help Moonshot? We built this in my academic group over the last few years. That then get integrated into my company Postera, into a product called Manifold, which is then offered for free to Moonshot, where basically we prioritize and generate synthetic routes for every crowdsourced designs. And that approach, using machine translation-based reaction predictions, we can then, once we have a good forward prediction model, we can go backward and basically do a tree search to find out which building blocks are actually, whether a molecule be made within, let's say, n steps from commercially available building blocks, and we can specify which building blocks we are allowed to use, Anamine's building blocks or Wuxi's building blocks, etc. And we can return a number of steps metric to guide us and medicinal chemists in the team which compounds are worth going after. So give you some examples of like why we think why machine learning approach, this approach actually I think achieves a speed up. I think one can always argue whether a machine learning based model knows chemistry better than a human chemist, and in fact I would argue, I would talk about a few slides later in the talk, that on paper very high performance of machine learning models can well be explained by dataset bias as well. But I think one thing that we find machine learning to be really powerful in solving is just the sheer problem of dataset size. For example, if you think about the number of building blocks available, I mean, in the good old days of Sigmund Aldrich, you might know like roughly what building blocks are available, and therefore you can just do hand disconnections. However, in the last few years, we have seen an explosion in commercially available building blocks, and even in ENEMI's database, they have over 100,000 highly functionalized building blocks available that you can disconnect to. And obviously, disconnecting to building blocks that are already on the shelf in Kiev is a much better idea than trying to ask people to make things from scratch. And this building block library is exponentially growing over time, and trying to just remember 100,000 building blocks is not going to happen. I think that's like a great time saver and opportunity that machine learning really seems to be able to accelerate and help. And there's a recent paper by an AstraZeneca a group showing that actually your simple amide coupling with sufficiently complex building blocks can already produce really complex, diverse, and new molecules. Again, showing that, you know, people are always, we are always like bemoaning how chemistry is constraining drug discovery, which is obviously true. But if we translate some of the hard chemistry into making complex building blocks, then when you're actually doing the synthesis, you can just use robust chemistry to couple these building blocks together. And that approach can well be sufficient to yield complex, novel, and diverse molecules. If you think about it, that's not dissimilar to how, you know, biology assembles complexity from 20 amino acids. Here goes a plethora of different structures. So if you have sufficiently complex building blocks, even the humble amide bond can, in principle, yield interesting molecules. To give you a concrete example of how this rapid triaging works in practice, the route on top is a route attempted by our CRO partner trying to make the molecule shown on the right. They start with a protection, then a urea formation, and then an N-arylation, which failed actually, and then they would deprotect. The problem is that the N-arylation failed, and even if it has succeeded, it will be a 4-step synthesis. Once we saw that, we put it into the algorithm, and actually the algorithm said, hey, an advanced building block is actually lying just on the shelves of their inventory. And so they can just literally pick up the advanced building block, couple them together. It's a 1-step synthesis, and you get the final material. And that was done and delivered in a week. These are really simple, you know, freshman chemistry. But again, the algorithm is able to rapidly decide which compounds are actually makeable using easy building blocks, which compounds are not. Another example where the CRO suggested this route on the top is a 5-step synthesis. They suggested it based on experience with a similar product. A molecular transformer suggested a 3-step route, which is a lot easier to execute. And again, the reason why we know that route existed is because we have access to real-time building blocks. And that's not a critique of, you know, human chemists not being able to keep track of building blocks, because building blocks are a funny object. They are constantly evolving, and there's no obvious pattern. Building blocks that are in stock today could be out of stock tomorrow, and vice versa. Like, for example, these three, I would say, you know, relatively similar building blocks, dichloro, bromo, chloro, and dibromo. One is available, $19 for 100 milligram. One is out of stock, and it's $451 per 100 milligram. And the other is like 4 to 6 weeks, and it's like $685 for like 1 gram. Vastly different prices, vastly different time scale to make. And there is no rule or pattern or whatnot. It just literally depends. I mean, I think we have depleted a lot of enemies building block, and now a lot of the Moonshot, the building blocks which we used to make Moonshot compounds are now out of stock. So the name of the game, I think, is that machine learning algorithms can, you know, rapidly be in sync with all these building block inventories, and therefore suggest dynamically which compounds are makeable, which compounds are not. And that's I think another reason why a lot of these synthetic accessibility scores you may see in the literature is not necessarily applicable, because what we need is not a gross synthetic accessibility, but synthetic accessibility is actually dynamically changing, evolving, depending on building block availability. Another thing about machine learning, which we have found out during the project, is that it is really good at doing mundane tasks. We got 2,000 submissions the first week, and you know, if we were to manually go through it, would take ages. But the algorithm can easily go through all the 2,000 submissions in less than a weekend, and find which compounds can be made with less than 3 steps. And if you go through one by one, it could take weeks. So again, really good at doing very mundane things quickly. And that's great, because you can quibble whether algorithms can make better molecules. I don't think it knows it has the creativity that chemists would have, but at least it can blindly go through compounds and with reasonable accuracy filter out the obvious bad actors. The next question we asked is, molecular transformer is great, but it's ultimately like a little bit unsatisfying that you throw, a language model of chemistry in and get a language model of chemistry out, just like Google Translate. Like, we know chemistry exists, we know physics exists, there must be something in it. A black box is quite unsatisfying. And then, so we took a step back and asked, what is unsatisfying about a black box? What do we want from how do we want to interpret a black box? And that really got us back to the drawing board to ask the question, well, suppose we want to interpret molecular transformer. What does it mean? And how should we do that? And we nucleated into sort of 2 ideas about interpretability and trying to understand what the model is actually doing. The first idea is, or 2 criteria actually. The first criteria is counterfactual, which is a reaction X occurs because of a certain functional group, which means that if functional group is not there, reaction X will not have occurred. The second criteria for interpretable models, we thought, is the idea of evidence synthesis. If a model reaches a particular prediction, that must be because of a certain data point. I mean, if you ask your undergraduate student to answer an Orgo question, you will want, first sketch out the mechanism, which is a counterfactual, and second, if the student is writing a term paper, then that student better cite some references. That's evidence synthesis. And I think a model that can do both would cease to become a black box. So we wanted to answer those 2 questions and really understand what the reaction prediction model is actually doing in practice. Seeing that we run this model many times, we see a lot of great predictions, but some predictions just catastrophically failed. So we say, well, is there like a scientific way of trying to understand these ML models? To give you a really short summary of what we did, we developed a way called input attribution, which is, remember I said in molecular transformer, we give the reactant and reagents input and predict the products. Well, suppose we consider a selective reaction. So reactions where a transformer predicts one product, but not the other. Well, can we attribute this probability difference back to selected functional groups in the reactants or in the reagents? If we can do that, then we can say, hey, are these attributions actually physical? We know how a reaction happened, physically, so we know roughly what should be the important things a physics-based or a model or a intelligent chemist should be looking at. So we can then cross-reference whether the model is looking at similarly physical parts of the molecule. Well, that's one idea. Another idea that we basically created is something called data attribution, which is a model in a very abstract sense takes the input, transform it into some model latent space, and then it spits out an output. So can we measure similarity between reactions, between the reaction you're asking the model to predict and any reaction in the database, in our data set, by thinking about the latent space distance between the reaction that we are trying to ask the model to predict and the reaction in the database. Again, this is not some reaction chemoinformatics tool, but it's literally asking, we open up the model, look at exactly the numerical weights and parameters that the model assigned to the input, and we look and we use that to look at which reactions are similar to which reactions according to the model. So we're looking at data attribution according to the model. So in this way, given a reaction, the model can say, hey, like, this is the adjacent reaction in the training set. And then we can see whether it makes sense or not. A really short summary of what we have done. So this is a epoxidation reaction. There are two alkenes. One alkene can get epoxidized over the other because one is more electron rich. We ask the model, hey, like you predicted one alkene get epoxidated rather than the other. Please tell me why. The model successfully attributed to the presence of two methyl groups, and that makes chemical sense. The methyl groups donate electrons into the olefin, making it more reactive. We validated with further examples. We can construct fake artificial examples of selectivity between a more or less substituted olefins, and we indeed show that the model does learn this electronic effect. If you think about input attribution, this is Diels order reaction. The model predicts something nonsensical. We say, hey, like, why did the model predict something stupid? So we, look at which reactions the model deemed the most similar, and lo and behold, those are not Diels order reactions at all. So of course, like if you have never seen a Diels order reaction, you will not predict a Diels order reaction. That's not bad. And this in general allows the user to actually understand, like if a model spits out an outcome, the user can actually ask the model, hey, like tell me why you made the outcome based on like which part of the molecule have you been looking at when you make the outcome to prediction, and which other reactions are you relying on to support this reasoning. And the user can then infer from this metadata whether the reaction is believable or not. Now, what is interesting is that we realize that actually if you ask the model to predict selective chemical transformations, the model can often predict things, the right products for the wrong reason. For example, if in a class of electrophilic aromatic substitutions where there are multiple different sites the molecule will react in, it turns out that we can arbitrarily bias the model to predict one substitution pattern by changing the number of substitution patterns the model sees in the training set. So effectively, the model can be fooled in predicting para substitutions. So two substituents being opposite to each other on a benzene ring by having a lot of para-forming reactions in the data set. And that has nothing to do with the electronic structure of the benzene ring. It just has something to do with there is a lot of para substitutions the model sees, and therefore whatever benzene rings you feed it, it will just shove the substituent in the para position. And we find that the model is actually intriguingly fooled by these data set bias. And these data set bias is actually quite prevalent in the literature. For example, the para substitution bias actually originates from the MedChem literature. There's a whole paper dedicated to this bias. And this actually started in the 70s because Toplitz, of Toplitz tree fame, a well-known guide to structure-activity relationship, actually suggested people, when they make a compound, always make the para-chloro and the para-methoxy as a way to probe structure-activity relationship. Therefore, there's a lot of compounds in literature with a para substitution pattern, and therefore that permeates in the data set, having a lot of para-forming reaction. And as such, transformer just, if you learn direct on data set, will just say, hey, whatever ring you give me, I will just put whatever para, and that would almost certainly be correct. Yeah, because the data set is biased. So we can fish out these like very interesting things about the data by really trying to unpack the black box. Just to quickly summarize, the Moonshot initiative has been going for now a year, and we hopefully show the open science crowdsourcing, this patent-free collaborative approach allows us to rapidly execute a fragment to lead campaign. Taking a step back, I think one of the learnings is that synthesis is incredibly rate limiting and also very costly. And that we see concrete potential in using machine learning to accelerate synthesis. by having access to evolving building block inventories. And I have not touched on that, but actually very simple library enumeration type ideas, which again, leveraging on synthesis, can work remarkably well in the hit to lead phase. So rather than thinking about complex synthesis, you can think about simple synthesis, but done in a parallelized library format. To end, I would like to thank my many collaborators. We wrote a bioarchive paper with over 100 people on it, and they all really contributed substantially to the project. And I would like to mention 5 collaborators which we meet frequently, almost weekly or more than weekly. Frank Wandel from Oxford near London, from Weitzman, John from MSKCC, Ed Griffin from MedChemica, a former med chemist in AstraZeneca, and Annette van Delft from Oxford. And them together with all our Moonshot network has been phenomenal in supporting this program, and we are very, very indebted to the support of the community and all our contributors. And with that, thank you very much for your attention, and we'll be very happy to answer any questions.
Thank you. That was a really wonderful talk. Please, those who have questions, if you want to unmute, please do so. And if you're shy, you can put the question in the chat and I can ask it as well.
Alpha, can I ask a question?
Sure.
Your approach goes against the dogma that pharma won't make drugs that they can't patent. So because they can't make their money back on their investment. So I'm wondering, how do you see the discoveries you have being followed through to actual clinical application?
That's a great question. I think in pharma, it's actually showing quite a lot of interest in the COVID space. I guess COVID might invite them to revisit this paradigm in helping open science efforts. And actually, folks like the NDI, the Initiative for Neglected Diseases, they have already pushed several drugs into market without any patents. And I think the way is twofold, actually. The first is, even without patents, there are other forms of intellectual property like market exclusivity that pharma can leverage. And second is that there's a lot of charitable organizations like Gates and the Wellcome Trust in the UK in this space, actually quite interested in starting to fund drug candidates in the IND enabling stage. So the message we got is that the discovery stage is hard without funding, but we are hopefully seeing a way to bridge that, and we do have some reserve funding to bridge that. But once we get to IND enabling studies and forward, there's actually a lot of national organizations, international charities willing to take this up together with charities like the NDI, LifeArc, and other medical charities to at least go to clinical trials.
Thank you.
Okay, I have a quick question. You motivated this program off of an incredible need to treat SARS-CoV-2. And through this, you developed a model and a system for drug discovery. And especially this, you focused on this prediction of synthesis. But another reason to do experiments rather than discovery as a way of testing or developing the model. So I'm curious, if you had a budget and you had to do experiments to better train your synthesis prediction, what would that look like, and how could you leverage your estimates of uncertainty, for instance, to focus in on things that your model doesn't know and would know better with better training data?
Oh, yeah, that's a great question. I think if we have budget, we would probably focus on like specific challenging reactions like Foucault-Hartwig, CN coupling, CC coupling, or other enabling reactions like Chen-Lam, Ullmann, and do a reaction screen of the different reactants against different solvents and ligands or whatnot, and collect that data so that the algorithm can really learn. I mean, rather than saying a Suzuki reaction shall work in this step, which after optimization may well work, the algorithm could say, this is the right condition, go run it. I think that would really help. And conversely, if after the reaction screening and Suzuki actually cannot work within the ligand space that is commercially available, then the algorithm would then have a piece of the negative data. We have been trying to leverage and sort of harvest all the reactions. In fact, we have a system of scraping all the reactions that have been run as part of Moonshot. But one thing we find is that in lab chemistry, often a failure, you don't really know whether it's a failure or it's just a different condition to make it work. And conversely, success could well be attributed to different factors. It may not necessarily be only the things that a chemist would write down, because there could be unknown factors contributing to that particular success. So I think probably the next step would be much more controlled settings where you focus on specifically hard reactions and then build a data set based on that to augment the machine learning model.
More questions?
So I have more questions about how your network can possibly fail. So is it possible for it to potentially present impossible chemistry, or do you have hard set rules on how to filter those reactions out? Or basically give us a clearer picture on what a non-accurate case would look like.
Yeah, that's a great question. A non-accurate case would be something like this, where it should produce all the reaction and the model just predicted some nonsense. And that is because it has never seen this reaction before. And with a attribution method, we can then understand that this is in fact the case. And in fact, the user would be would use this to say, hey, like this is the similar reaction from the literature. And you can look at it and say, hey, like, you know, these reactions are actually quite distinct or looks pretty dodgy. And therefore, you would have a feeling that this would be a bad reaction. In the retrosynthesis algorithm, though, we constrain the model to actually focus on robust organic reactions, at least in the hit finding stage. And so this constraint means that the model will not be at liberty to go for wild disconnection. So it may well predict the wrong product or the wrong isomer of the product, but it would not predict some wild chemistry because there is a constraint that there is only certain types of reaction, at least in the early stage of the project. In the late stage, we relax that and we get some more subtle selectivity issues potentially. But in the earlier stage, we constrain it to go for certain types of reactions like the standard 50 or 100 MET-chem transformations. And that means the model could mispredict the exact product, but it can't go for wild disconnections. I hope that answers your question.
Yes, it does. Thank you.
We're approaching the end of the hour, so let's thank Alpha again for this wonderful talk. Yay. And for the graduate students or others that want to stick around, Alpha has about a half hour to chat, and it would be great to get some of the graduate students to, to a chance to meet with Alpha.